Okay, so today we wanted to talk about the mode of action of pesticides. This is how uh, pesticides can work to produce an effect. Later on, we will talk about pesticide formulations. We will see how we are going to progress. Okay, I think you are now seeing the slide. Hello, can you see the slide?
Yes, Prof.
Okay, so we are going to start with the mode of action of pesticides. Now, basically, we will look at biochemical and physiological processes that lead to the destruction of the pest when it comes into contact with the pesticide. So, for this exercise, we are going to obviously we cannot cover all the uh, pesticides. So we'll just look, we're just going to use a few examples from organophosphates, carbamates, organochlorines, pyrethroids, and triazines. Now, as for fungicides, it's a, you may have to look at one of the notes slides which I sent you. It's just a short one on the fungicides. Uh, as far as organophosphates are concerned, these are widely used to control insect pests. They tended to replace organochlorines because of persistence problems with the organochlorines, which tended to stay for a long time and actually cause pollution, for example, DDT. But with the organophosphates, there's frequent sprays uh, which are used in order to control the pests during the growing season. Probably you may note that organophosphates are very effective on aphids. Now what you see where we have P, which is surrounded by Y, X, then we have got R1, O, and R2, O. R1 and R2 are representing methyl or ethyl groups which are attached to P. Then we have got Y. Y can be representing either oxygen or sulfur. Then X is a specific group on this molecule. So this is the basic structure of organophosphate. And it represents quite a number of compounds which are used as pesticides. So here we've got the methyl groups, CH3O, which is attached to P. We have got 2 of them. Then we have got either oxygen or sulfur attached to P, which is the central atom. Then on the other end, we still have that X representing other things, depending on the nature of the organophosphate. So on the organophosphates, the 2 other groups are usually methyl or ethyl and are the same in one molecule. X is a complex aliphatic. Now, X is also referred to as the leaving group. So you can have that structure POX or PSX. You can have either oxygen or sulfur within the organophosphates. Now let us have a look at the examples of organophosphates. Probably you may have come across some of these insecticides. The first group is known as the orthophosphates. What is key is that you have your central phosphorus atom joined to 4 oxygen atoms, and you have the other group representing other things there, either methyl or ethyl. Then X, the leaving group. These are some of the examples of insecticides represented by the orthophosphates. You have the clovenphos, you have got the dichlorophos, methenphos, then the phosphamidone. That's for orthophosphates. Then the thione phosphates or phosphorothionettes. Instead of having 4 oxygen atoms surrounding the phosphorus atom, please note we have got an S there representing the sulfur. Otherwise, the basic structure is the same. The examples under this structure, we have got the bromophos, diazinon, phenylthione, parathione. These are very toxic insecticides. Then primifos. We also have the phosphates or phosphorothioates. If I can move back, can you see where we have the sulfur there on the thione phosphates? We still have sulfur in the next group, but it is placed down and it is joined to the X. It's now down there. Once you put sulfur down there, you have a variety of these insecticides, or different types of these insecticides, the organophosphates. Examples, we have got the dimethyl S-methyl, oxydimethyl S-methyl, then we have got famidothione. Obviously, I don't expect you to memorize some of these names, but at least you have an idea about the structures. Phosphonates. Now with the phosphonates, you have got 3 oxygen atoms joined to the phosphorus, and you have got the X group, which is directly this time attached to the phosphorus. So we have got examples like triclophone and butonate. And this last one with a complex structure, pyrophosphoramines. Here we have got this kind of a structure. We have got something like a double P. You still have your oxygen atoms, but in addition you have the nitrogen atoms added to the phosphorus. And you have got those R2s joined to the nitrogen. So this is the kind of a structure of pyrophosphoramines. Shradan is the insecticide. So under organophosphates, we have got various insecticides depending on the molecular structure which is representing them. There are several enzymes which can attack organophosphates. The first group of enzymes are known as the phosphatases, or type A esterase, or just remember phosphatases. Then you have your cytochrome P450. Normally we refer to it as microsomal monooxygenase. When we are talking of microsomal monooxygenase, please note that we are referring to cytochrome P450. Then the other enzyme is carboxyl esterase. Then you have glutathione S-RR transferase. Probably there's a benzene ring somewhere. Then the glutathione S-alkyl transferase. This is where you have an aliphatic chain. I hope you understand the difference between a benzene ring and an aliphatic chain. But in any case, these are the enzymes which can actually break down organophosphates. Remember, we are trying to talk about the mode of action of organophosphates, how they actually cause damage to the pest, what sort of processes which are involved. This is the key issue. Now on that structure, the last one, where we have C2H5, that one can be affected by microsomal monoxygenase, right? Which is cytochrome P450. If you want, you can just say cytochrome P450 affect that area. Then somewhere in between oxygen and that group, carboxylase esterase can actually break that bond. Again, you have a carboxylamidase, which can affect the oxygen, which is pointed there. And also cytochrome P450 can affect the other end. If you want to call it microsomal oxygenase. Now, what is the mode of action of organophosphates? Basically, organophosphates affect the nervous system. I hope you understand when we talk of the nervous system of a pest, in this case, an insecticide. Now, there's an enzyme which is used during the transmission of a message in a nervous system. Nervous system actually transmits messages or signals, if you want to call them. What enables a nervous system to transmit signals is an enzyme called acetylcholine enzyme. The other substance which also helps to transmit the message is known as acetylcholine, or ACH. So within the nervous system, we have got acetylcholine, which can actually transmit a signal. But once it has transmitted a signal, it has to be stopped so that the transmission of the signal will be controlled. So you will now need acetylcholine enzyme to come in and destroy or and remove acetylcholine. So that is the purpose of the acetylcholine enzyme. It prevents acetylcholine from accumulating in the nerves. Because if this substance accumulates in the nerves, there is a continuous transmission of a signal. So what organophosphates do is that they are going to combine with the acetylcholine enzyme, because they sort of mimic acetylcholine. So when they combine with the acetylcholine enzyme, they form a permanent combination. Once you form a permanent combination, it means that there may be excess acetylcholine, because the function of the acetylcholine enzyme is to reduce accumulation of acetylcholine. But if its activity is interfered with by organophosphates, you now have excess acetylcholine. Once you have excess acetylcholine, there's continuous transmission of the signal, and this transmission of the signal will lead to the death of the insect. You can imagine if the insect was eating and it has been affected by this organophosphate, the insect will continue to eat. It will not stop until it dies. If it was moving, it will continue to move until it dies. It won't stop moving. So that is the activity of organophosphates. Here I'm just putting a picture of the nervous system so that you just have an idea of the nervous system. But you are really not required to draw this or to memorize this. So this is the area which is affected by the organophosphate. And we are talking about the nervous system within an insect. Even people may be affected if they get into contact with the organophosphates. Again, the full structure of the nervous system with all those names there. I'm not really going to bother you with most of these the names, dendrites, but these are simple, the biology. So this is how a nervous systems look like. The nervous system is supposed to transmit signals. It could be like electrical signals. This is like the wiring system of the living system. Where nervous system, where you have contacts during the, in the nervous system, you have got what is known as the synapses. In the synapses, this is where the pesticide may affect. That is where transmission is taking place. I will show you in a minute. Now on our left, we have the synapses contact. So normally you may have acetylcholine represented by those black squares, which can actually move from one area to the other and transmit a signal. After transmitting a signal, it goes back. Then in the middle, we have got acetylcholine and the enzyme. Acetylcholine enzyme, which actually reduces the levels of acetylcholine. On the far right, this is where you have, in fact, in the middle, you can see the combination of the enzyme CHE combining with the ACH. ACH is the acetylcholine. So it combines with the enzyme, but at a certain stage it will leave the acetylcholine. So the acetylcholine level remains low. But on the far right, where you have that red representing organophosphate, it can also represent the carbamates, which I will talk about shortly. So you can see in that area where you have the red triangle, which is joined to those stars which are green. The green star is representing the enzyme, and that enzyme is permanently joined to the insecticide. Once it is permanently joined to the insecticide, it can no longer control the levels or regulate the levels of acetylcholine. And as a result, there is a continuous transmission of the signal in the nerves. That continuous transmission of the signal will then kill the insect. Here I'm showing the actual molecules, but probably we don't need to spend much time on this. These are the actual acetylcholine molecules, how they interact with organophosphates. Probably this may not be clear, but the other one is clear. Now this one is just to demonstrate how organophosphate poisoning can occur, probably even in humans. You can see someone trying to spray. Now below there we are showing the nervous system, and in between we have got acetylcholine, ACH, right? Then we have got ACH E, which is the enzyme which sort of destroy excess acetylcholine so that it can stop the messages being transmitted. This is quite dramatic, this is a diagram, trying to dramatize what can happen. You can see that enzyme which is holding some knives, and it is actually, that is the organophosphate, right? Which is actually destroying the enzyme or making a permanent union with the enzyme. Once you reduce the enzyme, you have excess amount of acetylcholine in the nerves, and you have extreme levels of acetylcholine. So there's going to be overstimulation of the nerves. So this is a dramatization of the effects. If it happens to people, you get a paralysis. All sorts of things can happen during organophosphate poisoning. This is actually referring to the users or someone who is spraying organophosphate. Carbamates, another interesting group. The carbamates are derived from carbamic acid. Now there is a substance known as physostigmine or physostigmine, which is a natural occurring choline estrase. It occurs in plants. Currently, it can be found in calabar bean, Rhophysostigma benosas. This could be interesting. You know, some people who do what is known as witch hunting, they may actually use this substance, you know. They give it to people, to suspects, you know, so that they eat this calabar bean. It usually happens in West Africa, and probably even in Zimbabwe, I think so. Now, if somebody eats this calabar bean, the person may actually die. If the person dies, they will say, he's the one who has caused the witchcraft or something like that. But what really happens is that there could be excessive acetylcholine within the nerves, which actually kills the victim. And of course, people may respond differently. Some people will vomit, right? If somebody vomits, maybe the person is not guilty or something like that. I hope you understand what I'm trying to say. But the basic science is that this calabar bean is toxic. How it affects the victim is that it acts like a carbamate insecticide. It's like giving somebody a carbamate insecticide. So the carbamates are derived from carbamic acid. You have got carbaryl, carbofuran, primicab, also aldicab, oxamyl, methomyl, and so on. So these are the various carbamates. What these carbamates can do, they can affect the acetylcholine enzyme. Once they get into contact with that enzyme, they are not going to release it so that it can regulate acetylcholine. And of course, the acetylcholine will continue to give signals which end up killing the insect. So the mode of action of carbamates, they combine with the acetylcholine enzyme within the nerve tissues. Actually, the carbamates compete with the acetylcholine for the enzyme acetylcholine. So excess, please note, excess acetylcholine would cause continuous transmission of messages. It is this transmission of messages which will kill the pest. Remember, I said if the pest is eating, it will not stop eating. If it is moving, it will not stop moving until it uses a lot of energy, then it dies. So the mode of action is similar to organophosphates, but carbamates release acetylcholine faster than the organophosphates. So the carbamates are not as effective as organophosphates. Here on this chart, it's not, you are not supposed actually to remember all of this, but I'm trying to illustrate something. The first one, we have got acetylcholine in a normal system, and the acetylcholine enzyme. First, you have got a rapid reaction. If you combine these two, they will join together. Then they form something which is going to break down. And the choline is going to be released. And the enzyme is also going to be released. Then there is also a rapid reaction leading to a breakdown of that union. But if you go to carbamates, the first reaction, let's say with the carbaryl and acetylcholine enzyme, the first reaction is very fast, like what happens with normal acetylcholine. But the second reaction is very slow. This is the one which is going to lead to the accumulation of acetylcholine, and it will keep transmitting messages. It also same applies to organophosphates. The first reaction is rapid, but the second one is very slow. And the other one, it's either slow or it may be sort of a permanent feature. So this one will also lead to very serious effects on the pest. Now, as far as organochlorines are concerned, these are some of the widely used insecticides in history, especially DDT. You also have, other old insecticides like dieldrin, aldrin, heptadaclodin, linden, and toxaphene. These tend to stay in the environment for a long time. Well, these are some of the structures of the organochlorines. Now, the organochlorines, they affect the nervous system, but these ones, they affect the movement of potassium and sodium within the nervous system, and it is the movement of sodium and potassium which create some charges inside the nerve and outside the nerve. So we have neurotic effects which are caused by DDT type insecticides, which act primarily on the nervous system. They can act on the nerve muscle system. They destabilize neural activity. The result is that you have leakage of sodium ions through the nerve membranes, creating a destabilizing negative after potential. So this can cause, what is known as hyperexcitability of nerves. And this can actually kill the insects. Remember, the carbamates and the organophosphates affect the nervous system. But those two affect the areas where you have two systems coming together. But the organochlorines affect some parts of the nervous system. Here we just want to show you a normal signal during transmission, which is the first one, and the second graph is showing a series of those peaks indicating abnormality caused by organochlorines. Now, we have a group called the pyrethroids, which are obtained from chrysanthemum, Cyaneria folium, or you can just say chrysanthemum species, quite different names. These are actually grown in Kenya and Tanzania. This plant can produce pyrethroids, but the pyrethroids can be synthesized, to form insecticides. These are just examples of pyrethroids, various types which are used as insecticides. Now, the pyrethroids have got they resemble the DDT in terms of affecting the pests. They also affect the movement of potassium and sodium within the nerves. So they cause nerve excitation as a result of changes in nerve membrane permeabilities to sodium and potassium ions. These can cause rapid knockdown effects. They have neurotic action. As I have said, effects resemble those of DDT. They cause hyperexcitation and death. So sodium and potassium conductance are altered when the insect is poisoned. So it is the movement of the potassium and the sodium which affect the movement of the signals, and of course it can create something which is abnormal and cause the pest to die through excitation or hyperexcitation. Again, we can show this using those graphs. The first graph shows a normal transmission of a message, but the second one, there could be excitation due to the presence of the insecticide. So you have a different wavelength, as a result of the presence of the insecticide. People may use these signals to check the levels of poisoning in an organism. Here, it's a simple diagram of the nerve. The purple areas, this is where you have organochlorines like DDT and the pyrethroids affecting that nerve, where they affect movement of sodium and potassium. Where you have the nerves coming together, the area, the space in between, this is where you have organophosphates and the carbamates. What they do is to increase acetylcholine. Once you increase acetylcholine, then the signals are not stopped, and hence the insect pest may eventually die. Now we move on to herbicides. It's quite a mixture. Here on the herbicides, I've just taken an example from triazines. These are mainly soil-applied herbicides, but can also be used as post-emergence herbicides. Atrazine, which is in this group, is commonly used in maize as pre-emergence herbicide. Ametrine is normally used in sugarcane, and these herbicides can be persistent in soils. So these are some of the examples of triazines, right? Atrazine, simazine, etc. Again, different types of triazines. Yeah, I just want to, they have a basic similar structure which varies depending on what is added to them. The mode of action of triazines is that they affect photosynthesis. No wonder why the affected plants turn brown, especially the leaves. And of course, the plant eventually dies. So what they do is that they prevent electrons from the photosystem 2 from entering electron transport system of the chloroplast between the primary electron acceptor and the plastoquinone. I will show you the area where you have an electron acceptor and the plastoquinone where they block. So what happens is that there's now formation of active oxygen, leading to accumulation of peroxides. It is the peroxides which starts to burn the leaves. Also, nitrate reductase activity is reduced, and it can also lead to accumulation of nitrites, which also damage the leaves. Here I just want to say that atrazine will block carbon dioxide fixation. It will also block ATP generation. which are required for the dark reaction. So it can block CO2 fixation and ATP generation, as well as the NADPH generation. And it is the oxidation which will actually kill the plant and cause, these reactions will lead to plant death. So this is the message from this slide. All you need to remember is that it is the photosystem 2 which is affected. I'll show you the diagram. This is basically how photosynthesis works. I want you to focus on the top, the way we have primary electron acceptor, and the area where we have a plastoquinone. So the electrons from in between that area are stopped from flowing, which means everything down is actually affected. So that is the place which is blocked by atrazine or triazine. It's between primary electron acceptor and the plastoquinone in photosystem 2, which is to our left. Because photosystem 2 is depending on sunlight, which is hitting P680. On our right, we have got P700, and sunlight again hitting P700. So it is the left part, that is the primary electron acceptor and the plastoquinone, which is affected by triazines. So in summary, we can say that we have just looked at the mode of action of the following pesticides as examples: organophosphates, carbamates, organochlorines, pyrethroids, and the atrazine. So on the mode of action, this is it. We cannot deal with every pesticide on the mode of action. But what we should note is that once a pesticide enters an organism or a pest, there are some biochemical reactions which can take place, which can lead to problems in the pest. And of course, the pest will eventually die. So I will stop presentation at this point. Right. So this was the mode of action. Let me do something here.